You know, when we pop an antibiotic for a nasty cough or a fever, we usually just think of it as a standard, everyday pill. But today, in this explainer, we're going to look at what these medications truly are. We're diving into a silent, escalating microscopic war happening right inside our bodies, where antibiotics act as incredibly precise biophysical weapons designed to hunt down and dismantle bacterial machinery.
Comparing something like household bleach to antibiotics brilliantly illustrates the sheer elegance of these drugs. Think about what happens if you pour bleach on a bacterial culture. It's just violent, indiscriminate destruction, right? Bleach literally annihilates everything in its path through oxidation and protein denaturation. And the terrifying thing is, it would do the exact same thing to human cells. But a selective antibiotic? That's completely different. It enters your system, absorbs into your bloodstream by masquerading as dietary nutrients, and specifically hunts down bacteria at the site of infection, all without leaving a scratch on your own tissue. It's precision warfare, literally at the molecular level.
So how does that actually work? Well, it all comes down to a pharmacological principle called selective toxicity. Back in the late 19th century, Paul Ehrlich famously conceptualized this as a magic bullet. The whole idea hinges on exploiting the structural and biochemical differences between prokaryotic bacteria and us, eukaryotic humans. We are essentially using their unique biology against them, eradicating the pathogens while leaving our own cellular structures completely untouched.
Okay, let's dive right into this. Our narrative today maps out this war in 5 stages: the magic bullet, breaching the bacterial fortress, short-circuiting the membrane, the unexpected collateral damage, and finally, the evolution of resistance.
Stage 1, the magic bullet.
So what are the specific biological vulnerabilities that make this magic bullet possible in the first place? Basically, bacteria give us 4 massive therapeutic advantages. First up is the bacterial cell wall. Human cells completely lack this structure, which makes drugs like beta-lactams incredibly safe for us. Then there's the bacterial 70S ribosome targeted by tetracyclines. It's structurally entirely different from our own eukaryotic ribosomes. Third, bacteria have to synthesize their own folic acid from scratch, a pathway we easily block with sulfonamides, whereas we just casually absorb folic acid from our diet. And finally, their unique DNA replication enzymes can be totally jammed by fluoroquinolones. To put that into perspective, this is exactly why antiviral and antifungal drugs are so much harder on our bodies. Viruses replicate directly inside our own cells, and fungi share our eukaryotic structure. But bacteria? They hand us these 4 highly distinct targets on a silver platter.
Stage 2, breaching the bacterial fortress.
Let's zoom in on that first target, the cell wall. Think of this wall as a heavily armored fortress defending against some seriously intense internal pressure. You've probably heard the classic 1928 anecdote where Alexander Fleming comes back from vacation, looks at a petri dish, and finds a contaminating mold, Penicillium notatum, just aggressively obliterating neighboring Staphylococcus bacteria. But biophysically, what's actually happening there? Well, bacteria are packed with solutes, so water is constantly rushing into the cell via passive osmosis. This creates an internal turgor pressure that can literally reach several atmospheres. Without a rigid peptidoglycan scaffold to hold it all together, the membrane would just swell up and burst. This catastrophic mechanical collapse, which we call osmotic lysis, is the primary kill shot for cell wall inhibitors.
Now, building that protective scaffold is no small feat. It happens in 3 distinct phases, and the brilliant part is we can systematically sabotage every single one of them. First, deep in the cytoplasm, drugs like fosfomycin stop the bacteria from even creating the basic building blocks. But let's say a few blocks get made. Well, bacitracin steps in to trap the lipid carriers, trying to ferry those blocks across the membrane. And if they make it past that, drugs like penicillin swoop in to ruin the final cross-linking phase, physically preventing the wall from ever being completely built.
Let's move to how this builds into total structural failure. Beta-lactams are absolute master mimics. They disguise themselves as natural building blocks. The bacteria's cross-linking enzymes grab the drug by mistake, get permanently stuck, and become completely useless. Meanwhile, the bacterium's own maintenance enzymes are still casually trimming the existing wall to make room for growth. But since no new links are being formed to replace the cut ones, the structure rapidly weakens. All that immense internal water pressure we talked about forces the inner membrane to actually herniate through these unreinforced gaps. The result? Massive mechanical collapse and osmotic lysis. And just as a quick aside, gram-positive bacteria are extremely vulnerable to this because their thick wall is totally exposed, unlike gram-negative bacteria, which hide theirs behind an extra outer membrane.
Stage 3, short-circuiting the membrane. So, what happens if the bacteria are just sitting there, not actively growing? Or what if they're gram-negative and have that extra outer shield?
For those targets, we deploy polymyxins. You can think of these as microscopic electrostatic crowbars. That outer bacterial shield is tightly held together by stabilizing calcium and magnesium ions. Polymyxins come in and violently strip those stabilizing cations away. With the shield compromised, the drug jams its lipid tail right into the membrane, punching temporary pores that cause potassium to rapidly leak out. This sudden loss of power completely shorts out the bacterium, generating deadly oxidative stress that just tears the cell apart.
Now for gram-positive bacteria, we use daptomycin. This one is incredibly clever because it actually hijacks the physiological calcium naturally floating around in your body to switch itself on. When calcium binds to it, daptomycin drops its negative charge, exposes a hydrophobic tail, and wedges itself right into the bacterial membrane. Once inside, the molecules clump together, acting like powerful lipid magnets. They totally depolarize the cell and decouple the vital enzymes needed for cell division. The whole system basically freezes and shuts down, neutralizing the bacteria without ever actually popping or lysing the cell.
Stage 4, the collateral damage.
Let's zoom back out to the human body for a minute, because as insanely precise as these biophysical weapons are, friendly fire does happen. So the crucial point here is that our own evolutionary history actually betrays us a bit. According to the endosymbiotic theory, human mitochondria evolved from ancient prokaryotic bacteria. That means our cellular powerhouses contain structures eerily similar to bacterial targets. Because of this, certain antibiotics accidentally bind to our own mitochondrial factories. When that machinery stalls, it severely chokes our vital energy production, plummeting cellular ATP levels. In highly demanding tissues, this energy crash can trigger serious side effects, things like lactic acidosis, bone marrow suppression, and even neuropathy.
And the other massive area of friendly fire? Our gut microbiome. Broad-spectrum antibiotics are basically carpet bombs. They don't distinguish between a pathogenic invader and your friendly resident gut flora. To fix this devastation, we've historically been told to take probiotics. But a landmark 2019 clinical study out of Italy decided to put this to the test, comparing natural, unassisted gut recovery against multi-strain probiotic supplementation right after a round of antibiotics. Now, what's really interesting here is the massive plot twist. You would assume probiotics naturally accelerate recovery, right? Actually, scratch that. The study found that the group left totally untreated recovered their healthy, native microbiome in just 3 weeks. The group taking probiotics, it took them 6 months to get back to baseline. It's a wild dynamic called the probiotic paradox. Those introduced probiotic strains actually act as opportunistic invaders, aggressively hogging space in the gut and drastically delaying your native helpful bacteria from reclaiming their natural homes.
And finally, stage 5, the evolution of resistance. Which brings us to the final act of this story. Bacteria aren't just sitting ducks waiting to be destroyed. They're incredibly adaptable. They are constantly evolving these highly specific molecular counterattacks. Let's fire through how they handle polymyxins. Remember how those drugs rely entirely on electrostatic attraction to latch on to a negatively charged bacterial surface? Well, the bacteria just alter their electrical charge. They strategically slap positively charged molecules onto their exterior, neutralizing the surface and stopping the drug's magnetic attraction dead in its tracks. They even trade resistance genes with completely different species to pull this off. In the most extreme cases, some bacteria will undergo spontaneous mutations to shed their outer layer entirely, literally sacrificing a piece of their own anatomy just to survive the attack.
And it's an incredibly similar story for daptomycin and beta-lactams. Gram-positive bacteria will just mutate to lower their target lipid levels, effectively hiding from those lipid magnets we talked about earlier. Or they'll deploy specific molecules to artificially thicken their membranes, bouncing the drug away from their most vulnerable spots. Meanwhile, to fight off beta-lactams, bacteria frequently acquire the ability to pump out beta-lactamase enzymes. These are basically molecular scissors that just chew up incoming penicillins before they even have a chance to reach the cell wall.
So, what does this all mean? The entire magic bullet concept balances on a razor's edge of selective toxicity. We engineer these mind-blowing, highly precise biophysical weapons to exploit structural weaknesses. But in response, the bacteria just rebuilt. They refine their defenses, which leaves us with a truly provocative question as we wrap up this explainer. With bacteria constantly evolving new armor, strategically flipping their electrical charges, and swapping resistance genes like trading cards, who is ultimately going to win this microscopic biological war?
It's arguably one of the most critical questions for the future of modern medicine, and a huge reason why understanding these molecular mechanics is so absolutely crucial. Thanks for exploring this with me today.